This is the 14th video in the fundamentals of biology, and today we're talking about biotechnology. So one example of biotechnology is called DNA cloning, and this is the process of making many identical copies of a segment of DNA. The first step is to cut open a plasmid, and a plasmid is a circular segment of DNA that is contained within some organisms. And we cut it open using restriction enzymes. You would then insert the desired gene using DNA ligase, and this will become something called a recombinant plasmid. In the next step, we will insert the plasmid into the bacteria, and through transformation, bacteria will be able to pick up the foreign DNA. Because plasmids have an antibiotic resistant gene, we will use an antibiotic in order to eliminate the cells that did not pick up the plasmid. So we will have a colony that consists only of bacteria that have the antibiotic resistant with the plasmid. And finally, we will allow the bacteria colony with the plasmids to replicate, and by doing so, we will produce the desired proteins from the gene. So here's more on how to create a recombinant plasmid. So restriction enzyme will recognize and bind to restriction sites, and they will cut the double-stranded DNA in a certain pattern. For example, something called EcoRI will create a short segment that overhangs. So in this picture, the EcoRI enzyme will cut along this G on both sides, and as a result, there will be sticky ends because another segment cut by EcoRI will be able to connect to its ends. So contrary to EcoRI, restriction enzymes that only cut down the middle will create blunt ends, and they are harder to connect back together. Because EcoRI will cut a plasmid and a target gene, they are able to connect together through DNA ligase. So you can see in this picture that a target gene is cut in these locations, whereas the plasmid is also cut down this middle, and as a result, the target gene can enter into the plasmid. And finally, ligase will seal this gap together. The next type of biotechnology is the polymerase chain reaction, or the PCR. And this is when we make copies of DNA in vitro, or outside of an organism. The first step of the PCR is to denature the DNA, and this is when we apply heat in order to separate DNA strands in order to make a single strand template. So in this picture, you will see that we will raise the temperature to 96 degrees Celsius in order to denature the DNA. The next step is annealing, and this is when we lower the temperature so a DNA primer is able to begin adding complementary strands. So in this picture, we lower the temperature to 55 degrees Celsius, and similar to DNA replication, we will require a primer to begin adding the strands. Finally, in extension, we will re-raise the temperature so that a polymerase called Taq polymerase will be able to extend these primers, similar to DNA polymerase in replication. This will result in a copy of a DNA, and we will repeat this multiple times. Gel electrophoresis is a process to separate DNA fragments based on their size. So here is some basic terminology for the parts involved in gel electrophoresis. So an agarose is a polysaccharide gel in order for DNA to pass through. The gel box holds the agarose, and one end will be negatively charged and the other is positively charged, and it is charged through a power source. And the wells are indentations on the negative charge for the DNA samples to be placed. So in the first step, we will insert the DNA samples into the well, and we will allocate the first one as a standard reference called the DNA ladder. So in this right picture, we will put the DNA samples in the right wells, allocating the first one for reference. In the next step, we will turn on the electrical power from the power source. And since DNA is negatively charged due to the phosphate groups in DNA, the fragments will move towards the positive end because opposite charges will attract. In the next step, as it runs, shorter fragments will travel farther, whereas longer fragments will travel less distance. In using UV light to observe, the bands created can be compared to the DNA ladder, to determine the presence of certain DNA fragments. The final type of biotechnology is called DNA sequencing, and this is used to determine the sequence of nucleotides in the DNA. One method of DNA sequencing is called the Sanger sequencing or the chain termination method, and this is especially useful for short sequences of DNA. Some things required for DNA sequencing using the Sanger sequencing method is DNA polymerase, a primer, the 4 DNA nucleotides, as well as a template DNA. But along with these things, you will also need something called the dideoxy version of these nucleotides. These versions of the nucleotide have no hydroxyl group on the third carbon. So in this top picture, you can see that the third carbon, 1, 2, 3, this carbon does not have an oxygen, so it is not a hydroxyl group. And what this does is it prevents the connecting of nucleotides. So that's why we call it chain terminating. And each of these dideoxy versions are marked with a different color of dye. To prepare, we will create a sample containing primer, polymerase, the nucleotides, as well as a small amount of the dideoxy nucleotides. So first we have to replicate the DNA. So as we did in PCR, we will heat the sample to denature the DNA, we will cool it to let the primer build, and we will raise the temperature again to let polymerase synthesize the complementary strand. As we replicate all of this DNA, sometimes a dideoxynucleotide will be added instead of a normal one, and because it was chain terminating, it will end the strand with a marked nucleotide. After many cycles, it is almost guaranteed that every single position of the target DNA has been terminated with a marked nucleotide, since we have many samples of DNA being used. Finally, the fragments are run through capillary gel electrophoresis, and using a laser, the dye at each fragment length is detected. And using a chromatogram, we will be able to observe the dyes detected. And using this chromatogram, we will be able to find a specific sequence of the DNA. So thanks for watching. This lesson covered DNA cloning, PCR, gel electrophoresis, and DNA sequencing.